All right, so this is on 3.6, kind of on 3.6. It's about enzymes. I actually wanted to talk to you a little bit more about enzymes and give you some context for these enzymes and connect it really back to the first couple of sections of this unit, 3.1 and 3.2, as well as the previous chapter and previous unit on body systems. So we're going to talk a little bit about enzymes, but I actually want to start at the back of this section, near the end of that section in your book. One of the examples that you are expected to know is about lactose intolerance. So I'm just going to jot that down and then tell you a little bit about lactose intolerance. And I'm sure that you probably know a little bit about lactose intolerance, but this is simply the idea that lactose sugar can't be digested. Now, not everyone is lactose intolerant, but some people are, and in some cultures, almost everybody or everybody is, and in some cultures, almost everybody is not. And we'll talk a little bit about why that is, and what's the mechanism going on here. But you should, just as a quick note, you should be able to recognize that lactose is a sugar because of the OSE ending. So think back to the other examples from a previous video and discussions in class, and that you should know what the word digestion means. And I'm not going to define it again, but this should jog your memory a little bit back to the digestive system and the definition of digestion. So this lactose can't be digested, and lactose is a sugar found in milk. It's found in milk or dairy products. So this lactose can't be broken down. And really what it comes down to is a fairly simple reaction in which the lactose sugar, I guess I just use a different color pen here. Lactose sugar plus water plus the enzyme that we call lactase. Remember, the enzyme is often named after its substrate, so the enzyme here is lactase, its substrate is lactose, and what it does is breaks that lactose up into galactose plus glucose, and this is the reaction. And so I'm not going to tell you, I want you to figure out what kind of reaction is this. Is this a condensation reaction or hydrolysis reaction? And I'm going to push you back to look at your notes from a previous video or from your notes from 3.6 and see if you can make sense from this. People who are lactose intolerant have a problem with this enzyme. So people who are lactose intolerant have, I'll simply say it this way, a different version of lactase, which prevents the digestion of lactose. So we need to talk a little bit more about what that really means. So I'm just going to move down a little bit. So a different version of lactase. Now remember that lactase is a type of enzyme, and enzymes are a type of protein, or sometimes they're referred to as polypeptides. And proteins are made of amino acids. So you might know something about amino acids. I hope you do. Some background knowledge as well as from a previous video about the structure of an amino acid of the NCC background. It's got a carboxyl group, an amino group, and a R group or variable group, or that R stands for actually residue group. So I'm just going to draw a generic model of a protein. It's very simple. It's not going to show you much detail. But here, let's say that this purple sphere represents one amino acid. Got a second amino acid, a third amino acid, fourth amino acid, fifth amino acid. It keeps going for hundreds and hundreds of amino acids in this direction. And let's say that this is the start. Let's just say that this is the start for our model. When these amino acids are bonded together, hundreds of amino acids long, we get a protein. When it's a specific order of amino acids, just like to make the word protein, I have to put these letters together in the right way and not put in any extra letters. Enzymes and proteins work the same way. It's got to have the right amino acids in the correct order. Because if you remember this structure, here's my carboxyl group, here's my R group, here's my H group. Well, it's this R group that actually gives each amino acid, so it's the R group, that gives each amino acid unique, I'll say fairly unique, chemical properties. So some of those R groups, let's say some of these R groups could be hydrophobic, which I think you know what that means now, based on the polarity of water. So this would mean that that would mean that these molecules are non-polar. They could be alternatively I'm sorry for writing so small, hydrophilic, which means polar. They could be acidic, or they could be basic. And really what that means is that these amino acids are going to interact with each other and their environment very differently. Remember that all proteins are made by living things. So we've talked about viruses before, and viruses don't have their own metabolism. They don't have their own metabolism because they lack ribosomes. They're so small the ribosomes can't fit inside of them, and it's ribosomes that put these amino acids together in the order they are and make those proteins. So all proteins are made by living things, and remember that all living things have lots and lots of water in them. And so this is really where the properties of water, so the properties of water, really coming back to the polarity of water, I'll say shape the protein. So coming back to this linear model of protein, a sequence of amino acids, well, this is the most basic level of structure for an amino acid. In fact, we call it the primary structure. But when we start to think about the R groups that are coming off the top of each of these amino acids, well, they're going to interact with the environment in very different ways. If they're hydrophobic, they're going to want to fold in on themselves. Think about this in a three-dimensional shape. They're going to fold in so that they're not touching any of the water outside of the protein. And the hydrophilic amino acids are going to be touching the water. So this is going to fold up into some, unique three-dimensional shape. That's my scribble, but imagine that in 3 dimensions. It's going to fold up in some three-dimensional shape, which is really important because think about the active site and substrate interaction. So we're talking about lactose and lactase interacting. We're talking about that in a three-dimensional way. So if the pocket, what we call the binding pocket, where the active site is, changes shape, it won't be able to latch onto the lactose. So we need to talk about how is it, let's see if I can grab okay. So let me pick a more interesting color here. So how do proteins change shape? Well, when we talk about changing shape, if it's environmental, We call it denaturation, or we say that the protein is denatured. And really, the way that that's happening is if we increase the temperature or change the pH, or, and this is vague, but certain chemicals can interact with those amino acids, changing the environment in which they are forming and taking shape and changing their shape. So that's denaturation. But alternatively, and what's going on here with the lactose intolerance is a genetic mutation. Or maybe I should just say an alteration. Because you might have some connotations or misconceptions about what mutation means, but essentially these amino acids, I think, let me scroll down. These amino acids are put together in a very specific way, and that's very important. And those amino acids are put together because of a set of instructions that were sent to the ribosome to put these amino acids in this order. So if those instructions get changed, we end up with different amino acid sequences. So a change in DNA, or the instructions, leads to changes in amino acid sequence. Now, if the amino acid sequence changes, it also changes the way that protein, the polypeptide, is going to interact with each other. And that's problematic. Oh, by the way, I think I forgot to mention this earlier in a previous podcast, and I just wanted to give you this little nugget of information. These bonds between these amino acids, as you know, the amino acids were put together through the process that we refer to as assimilation in the digestive system section. But when we put little pieces together, you should recognize it as a condensation reaction. But for proteins specifically, each of these bonds are called peptide bonds. So when we have a lot of amino acids with lots and lots of peptide bonds, we call it a polypeptide. Okay? Poly meaning many. So, in essence, if someone has a genetic alteration that makes a slightly different version of the protein, then that protein is not going to be able to interact with the substrate lactose and not break it down. And I apologize, I forgot to mention this. This is a disaccharide, lactose. So when disaccharide gets broken up, you should be able to predict that galactose is a type of monosaccharide and glucose is a type of monosaccharide. I'm just abbreviating it as mono, not writing out saccharide. Okay, so that's essentially what's going on with lactose intolerance. So the way that we use biotechnology to help us with this is that we add we'll call it lactose-free milk. You might have seen this in the store. You might drink it yourself. You might have someone in your home who drinks it. Lactose-free milk is milk in which lactase, usually it's from bacteria that we use this. So it's a protein that bacteria also make, and we grow in the lab. That lactase is added to milk before they put it on the shelf at the grocery store. And by adding lactase to the milk, even though we're people who don't naturally make lactase and go through this reaction in green, this reaction is already happening in the milk supply before they get it. So people who are lactose intolerant aren't worried about digesting lactose. They've already got these two monosaccharides in the milk supply, galactose and glucose, and because they're already monosaccharides, they don't need to digest them, even though it's a different kind of sugar. They don't have to digest them because they're already monosaccharides and will be able to be absorbed across the membrane of the villi into the capillary beds and be transported through the bloodstream. Okay, so I hope that makes a little more sense, but I want to come back to this idea of denaturation because there are a couple of things that we need to know, and one of these things is exactly how is it that enzymes work. And what enzymes do, how do they work? Well, there are a couple things to know about enzymes. They are catalysts, which mean they speed up reactions. They can be reused over and over unless they become denatured, in which we have to make more of them. They are organic molecules, specifically proteins. Okay? And so there are a couple of things that, and as catalysts, they speed up the reaction by reducing what we call the activation energy, or the amount of energy to start a reaction. So by reducing the amount of energy that's required, the reaction moves forward faster at biologically active rates. If we didn't have these catalysts, we wouldn't be alive today. So there are a couple of factors that affect the rate of the reaction. We talked about some of them, and one is temperature. We'll talk about that again. 2 is pH, and 3 is substrate concentration. And there are graphs that go along with each of these that help explain what's going on. So we'll draw these, talk about these, and I think call it a day. Okay? So there are a couple things that you should be able to know already. Be able to define enzyme and active sites, explaining enzyme substrate specificity. Now we're talking about 3.6.3. When we talk about temperature, really what you need to be able to do is annotate the graph that you have on page 76 of your textbook, which looks something like this. Well, actually, maybe this part is a little more straight. Okay, and so make sure that you label your axes. So temperature increases as we move to the right, and then the rate of the enzyme reaction. So I'm just abbreviating a couple things. So I'm going to label a couple things and verbally annotate this graph. I hope that you jot down these annotations. So as temperature increases, if you know what temperature is, it's a measurement of how fast the molecules are moving. It's a measurement of energy. So here, as the temperature increases early on in the graph, here in A, we understand, well, I hope you understand, the molecules are colliding more. They're running into each other even more. So enzyme and its substrate are finding each other much more frequently. If they were standing still, because there's a zero Kelvin temperature, then they wouldn't be interacting at all, and we would have no reaction at all. But if we speed up the reactions by increasing the temperature, those molecules are more likely to bump into each other and drive the reaction forward. But here, at B, at the very top, the enzyme starts to lose its shape because the temperature has gone beyond its optimal temperature. And when it changes its shape, it's no longer going to be able to speed up the reaction of the enzyme and substrate reaction. And here in C, the increased temperature continues to change the shape of the enzymes, so even increasing the temperature beyond the optimal temperature is actually going to drive the reaction rate down, because you're taking enzymes out of commission, essentially, and so they're no longer able to bond with the substrate to break it down or build it up. So there's an optimal temperature, and I hope you know that if we're only concerned about the human body, our body temperature is about 98.6. That is our optimal temperature for all enzymes in our body. All right, so let's talk about pH. pH is a little bit different, and I want you to think about some of the enzymes that we talked about in the digestive system unit. So you know that the digestive system has several different enzymes with different optimal pHs. We've got amylases, we've got lipases, we've got proteases. So we're going to look at, we'll label pH at the bottom. We've got neutral here, low pH here, high pH here. So low pH means acidic, high pH means basic. And then again we've got I'm trying to write this sideways as best I can. The rate of the enzyme substrate reaction, and it depends on really on which enzyme we're talking about. So if we were going to map, let's say, proteases that are in the stomach, well, we know the environment in the stomach is highly acidic, so it looks something like this. It's a very rough a graph, but you need to understand this. Or if we looked at the amylases, especially the salivary amylase, we would see something like this. We don't really see too many that are on the basic end for us. And so really what you should understand is that there are different levels of optimal pHs for different enzymes. And the reason for this is that if you know what pH actually measures, pH is a measurement of hydrogen ions, a concentration of hydrogen ions. Well, hydrogen ions are going to interact with polar molecules, and if you think back to that one of the R groups, one of the characteristics of the R groups rather, is whether it's polar or not. And so these hydrogen ions are going to interact with those amino acid residues, or the R groups of those amino acids, and change the chemical properties in the way that they interact with each other, thereby denaturing them. So there is optimal pH in different parts of our body, and there are buffers, and I hope you learned about buffers in chemistry, but buffers are chemicals that, without going into too much detail, help maintain the pH in a certain area of your body so that it doesn't change drastically, because as you can imagine, if it changes drastically, the enzymes and all the other proteins become denatured, and you have to wait for your body to make new proteins out of the DNA instructions rather than having them on hand. Now, the substrate concentration one is a little different. So substrate concentration, so let's put that down here, substrate concentration, and again, rate of reaction. Now, what you should know is that the substrate concentration graph looks something like this. And the reason is, when we're thinking about collisions, and we think, let's imagine a beaker, and if I put in so much substrate, well, let's say I put in so much enzyme. I'm not changing enzyme in this graph. The amount of enzyme is the same. Okay? So here's my water line. So it's in the aqueous solution. We've got our enzymes. I'm not changing that, but as I add more and more substrates for the enzymes to work on, the rate's going to increase. And that's really exactly what's happening here at this slope. But at a certain point, there's only going to be so many enzymes that can work on a substrate at a time, so the rate of the reaction is not going to increase or decrease unless we change the shape of the enzymes by temperature or pH. But the reaction still continues. So the reaction is still going on at this level, but the rate's not changing. So we don't see that line really increasing or decreasing because we've only gotten so many substrates. So think about this: like if I were to ask you to text your mom in class, and there are 30 of you in class, then that's the fastest that you could go. But if I reduce the number of enzymes or phones that are accessible, so if I reduce it down to 2 phones, would the reaction rate go as quick? Would it take everybody longer to text their mom if they only had 2 phones from which they could text? Yeah, because you can only use 2 at a time. If you increase the number of substrates, then the reaction rate can increase. Up to a certain point, but this reaction levels out. What you could do to increase the rate even more is to add even more enzyme to work with the increased amount of concentration, and you would see the rate increase. Okay, and I hope that makes sense. But I want to go back to the lactose intolerance very, very quickly, and I mentioned that this is my closing question for you. That some populations are lactose intolerant and some aren't. So, and it's based on DNA, essentially instructions that code for a certain version of the protein. So let me ask you, see if you can figure this out. So this is based on what you know, and understand about evolution and natural selection. If you think they're the same thing, then you might need to do a little bit of research. Evolution and natural selection, based on what you know about those things, what kind of evolutionary advantage did lactose tolerance in adults have over those who did not have that genetic alteration? So again, this is due to a mutation. We know that. Remember that humans are the only population of organisms that consume milk as adults, and we drink it from another species most of the time. So, as adults, we do. So why is that the case, and what evolutionary advantage did it provide people? So much so that in many cultures, if lactose intolerance is in the gene pool, oftentimes it's prevalent and outnumbers those who are lactose intolerant. Okay? So think about that, see if you can come up with an answer. All right? And I think that's it.